As I see that we are now admitting our attendees, let me welcome everyone who is joining and who is participating and watching us. We are back at the webinar series of the fruit flies in silico prevention and management project under the Horizon 2020 European Funding for Research and Innovation. Today we have a different topic that we will focus on, and today we will discuss about odorant-based detection of fruit fly infested fruits in cargo shipment. We will hear more about our topic later on. Please have in mind that we intend to record this webinar for future dissemination actions of our project. And it is important that if any one of our attendees needs to send us a question, that you can use the Q&A button for questions later, as our presenter will give us enough time to have questions and receive whatever you need to ask and provide you with his answers. I think that we can move forward and start. I will ask our moderator and project coordinator, Professor Nikos Papadopoulos, from Department of Agriculture, Crop Production and Rural Environment of University of Thessaly, to briefly introduce us to today's topic, and to our speaker, Dr. Panagiotis Milonas. Thank you, Professor, and we are here to hear you. Thank you.
Thank you very much, Georgia, for the introduction, and good morning to the webinar of the FF-IPM project. This is the 5th webinar. In fact, our project is heading to the end, and we are concluding a lot of studies, and we are happy to share with all of you some of the results of these studies. So the topic of the webinar of today's webinar is the interception of infested fruits in cargo shipments. This is a really challenging aspect in all this process of the invasion of fruit flies and of other pests in Europe and in other parts of the planet. So the seminar is given by a prominent entomologist, Panos Milonas. Panos is currently a head of the entomology unit at the Benaki Phytopathological Institute, but he has a rather long history in fruit fly and pest management, starting from the Aristotle University of Thessaloniki, then spending some short time in private companies dealing with agriculture and management of pests, and then spending some time in the States working on behavioral ecology, and is currently involved in many projects funded by the European Union, by other organizations. Also, he is collaborating for some period of time with EFSA, serving on the panel of EFSA. So his experience is really long, and the target of his recent efforts on detecting infested fruits based on odor is, I think, seminal. So we are really looking forward to listen to talk of Panos Milonas. Panos, the floor is yours.
Good afternoon, Nikos. I hope that everybody can hear me. I had some issue previously when you were talking. Thank you for the introduction and the nice words. I guess I need to share the screen now. Should be okay.
It is.
Okay, thank you for the confirmation, Georgia. So good afternoon to everybody, to all FF-IPM friends who are viewing this webinar today, which is a hot day for Greece. So a special thank to the Greek colleagues, because they are here listening to us instead of being for swimming, which is the first hottest day for the summer. So as the title indicates, I will try to provide some information about the possibility of detecting infested fruit commodities, which are infested by fruit flies based on their odors that they are emitted by the fruit itself. Okay. briefly to outline the presentations that I'm going to do today. Just a very quick touch on the plant defense mechanism. What are the herbivore-induced plant volatiles, and what is known as the theory for cry for help. What do we know about fruit flies and herbivore-induced plant volatiles? And then I'll show you our results through the FF-IPM project about the herbivore-induced plant volatiles on certain fruits infested by fruit flies. And finally, the concept of electronic nose and the results that we have from the FF-IPM project. So I'm sure that we all know that the plants are sessile organisms and are rooted in the soil, and they face a lot of enemies. So there are large enemies that are eating them. And the most diverse taxon of attractors consists of insects. However, plants are far from being passive victims of these attractors, and they can activate a multitude of defense responses. And direct plant defense, which has to do with the characteristics of the plant that affect the attacker, but also other defense strategies that they have to combat mobile attackers is to enlist bodyguards such as insect predators and parasitoids, or endomophagous nematodes, or even insectivorous birds. And this can be done by providing shelter or alternative food, or through the emission of the herbivore-induced plant volatiles. And plants actually communicate extensively with other organisms in the environment through these volatiles, through volatiles in general. And some of the volatiles can be induced by the herbivore, by other herbivores feeding on the plant. And the induced emission of plant volatiles that attract carnivorous organisms, predators or natural enemies, is a phenomenon which is oftentimes referred as crying for help from the plants. And it has been demonstrated for plants in several plant families and occurs in response to a wide variety of herbivorous insects and mites. The induction of bodyguard attracting plant volatiles was first demonstrated for mites, and then quite soon for insects since the late 1990s. But later it was reported as a response to feeding by a range of the group of insects. And now we know that plants may also emit and initiate emission of induced volatiles in response to oviposition by herbivorous insects, what is known as oviposition-induced plant volatiles, or even in response to exposure to induced volatiles from herbivore-infested neighborhood plants. So carnivores are attracted to these volatiles from different orders, and some studies have shown that there is a positive correlation between the amount of the released volatiles and the degree of the response. However, when this was studied specifically in a plant insects combination, it was clear that the quality of the herbivore-induced plant volatile blend was as important as its quantity. And indeed, we know that not all of the constituents of the blend that is emitted elicit a response by the carnivores. And we need specific electrophysiology studies like gas chromatography, electroadenography to reveal which compounds within the blend of the blend emitted elicit a response in the chemoreceptors of the carnivore. And actually, such studies have shown that the compounds that actually dominate the blend are not necessarily the most important ones for the carnivores, which is not surprising given that the carnivores must evaluate how reliably the compound predicts the presence of a feeding herbivore. The emission of the herbivore-induced plant volatiles occur locally at the site of infestation, but can occur also systematically from uninfested leaves. And the carnivores that search for the herbivores actually face a big problem, because their victim is under natural selection to minimize the emission of cues that guide the carnivore to them. So, and however, the herbivore-induced plant volatiles emitted by the plants from which the herbivores feed can solve this problem, because they are emitted in relatively large amounts and the composition of the mixture can be herbivore specific. So we have a source which is the herbivore itself and is of high reliability but of low detectability. And through this combination of the herbivore-induced plant volatiles, the carnivores minimize this problem. And the herbivore-induced plant volatiles are both detectable and relatively reliable cues that can be exploited by the predators and natural enemies. And we know now that a lot of insects in these groups have been found to respond to these volatiles. And actually, they are much more sensitive than the analytical equipment is. And carnivores can often discriminate between plant volatiles induced by different herbivore species. And the blend composition indeed is herbivore species specific, which is very important and actually guided also our research within the IPM project. Now, a little more about the theory. The attack of an herbivore results in a large-scale transcriptional rearrangement in the attacked plant. And this includes the induction of genes involved in phytohormone biosynthesis and the biosynthesis of secondary metabolites. The volatile blends, the induced volatile blends, can comprise tens up to hundreds of components, as we will see in a while. Thus, a herbivore-infested plant emits complex blend of volatile compounds. And these include, for example, terpenoids, green leaf volatiles, aromatic compounds, and others. However, only a subset of this complex mix of the components mediates the attraction to the receiving insects, as I already say. And to identify which compounds are involved in the attraction, that can be done by individual compounds or by interference with their interaction or biosynthesis in the plant. There are also elicitors. The elicitors for herbivore-induced plant volatile induction are present in herbivore oral secretions, and application of oral secretion or the elicitors have shown that can mimic the effect of the herbivore and induce the activation of single transduction pathways. So the induction of the herbivore-induced plant volatiles is mediated by phytohormones such as the jasmonic acid, the ethylene, and salicylic acid. Moving forward now to the problems that we are facing, the detection of pest infestation in fresh produce that traded internationally could offer improved prospects for reducing the movement of unwanted pests, because immature stages of some pests can be difficult to find visually. Other cues such as herbivore-induced volatiles that I just talk about can potentially be detected especially at the early stages of the infestation, and it's worth investigating them. And in the family of the Tephritida, the fruit flies, the true fruit flies, contains a large number of species that are pests of fruits, and they require intensive management and surveillance to maintain market access for produce. So due to this, their economic impact, much attention focused on detection and monitoring of adult flight populations. Their larvae, we know that their larvae feed and develop while hiding within the fruit pulp, making infestation difficult to be detected by inspections and inspectors, especially eggs and first instar larvae, which are clear to pale white in color and camouflage very well within the fruit pulp. infestation by fruit flies at early stages, therefore, can be undetectable. So agricultural inspectors oftentimes should perform destructive sampling in a large proportion of the commodities that are being inspected. And when a sample has been taken from the consignment because it's suspicious for pests present, then the fruit remains under official control and cannot be released until the final laboratory result confirms that there is no quarantine pests in there. So there is absolutely need for sensitive and fast screening for larval infestation. So here, we actually want to take advantage of the response of the plants to herbivore, which is done for their help to attract the natural enemies in order to detect infested fruits. And we are not the first who thought about that. There are studies in the past where the volatile organic compounds have been used for detection or differentiation of infestation from different tephritid fruit flies and various host fruits, as you can see on the slide. But most of these studies that use a technique which is called solid phase microextraction, where only the last one, which was on grapefruit, uses the 9 head space technique that we also use in our studies. I will explain that in the methods ahead. Well, in these studies, especially one of them found that there is a difference in the total amount of the volatiles emitted in the infested fruit, apples in particular, apples infested by Queensland fruit fly, Bactrocera trioni, and as you can see, there are actually here there's a difference. There are less amounts emitted by the infested fruits. So within FF-IPM, we all know that there are 3 fruit fly species that are studied. Ceratitis capitata, which is well known and has been established in the Mediterranean coastal and other areas, but also responding to climate warning that the relaxed climatic barriers, population have gradually spread to northern areas in Europe, which previously were thought that they are too cold to allow population persistence. And Bactrocera dorsalis and Bactrocera zonata, which are not yet established in mainland Europe, but are rapidly spreading towards Europe. So the aim here is of our studies was to determine infestation-specific volatile compounds that can be indicators of infestation by the 3 different fruit fly species: Ceratitis capitata, Bactrocera dorsalis, and zonata. But then with the ultimate goal to develop a rapid, reliable, and cost-effective method to in detecting this in infested fruits, reducing the time that is required for inspection and to avoid the unnecessary destructive sampling, which is currently the common procedure. A few words about our methods. Well, first, we wanted to achieve infestation of the fruits that we used. And for this, we were fortunate to have laboratory readings of the fruit species here in our institute. And then we present fruits to a group of adults in a cage. And after a certain period of time, the fruits were inspected for reposition. And then the fruits are stored in specific growth containers for different periods of time, depending on the fruit and the temperature that they use to maintain the fruit. And at different time intervals, we collect the volatiles. And you can see how we do that in the pictures both to the right. And to the top left, you can see the number of samples that you use to take volatiles from the different fruits. We examined fruits samples from peaches, from pears, apples, and oranges. And you can see there are more than 500 samples in total that were examined. So here, a few words about the technique. We call it dynamic heat space technique, contrast to other static techniques. Here, there is air that is pumped through the container, which is a glass container, as you can see, where you have the different fruits inside. So we have charcoal filtered air going through, and also the air is pumped out continuously. And at the exit, there is an absorbent who specifically uses Poropac. And then there the volatiles are trapped, and then this will take the sample from there, elute it, and then goes for GCMS analysis. And for quantification, we use external standard calibration and several statistics that then takes place for analyzing the results. So directly into results, you can see here some chromatographs from the pitch trials, and you can see the different peaks of the compounds that they were identified. And specifically from pitch, we identified 85 compounds in non-infested fruits. And you can see here the level of amount of the different compounds, linalool in particular, and ethyloctanoate, an unknown compound here, which change as time passes, as you can see in these chromatographs. And now linalool is much slower than it was in the beginning of the experiment. And in total, in the infested fruits, we have found 87 compounds, so 2 more than was not found in the non-infested ones. Again, here you can see the differences of some compounds in comparison to the previous chromatograph in respect to the abundance of that. If you see it in total as group of compounds, there is this increase with time for esters and lactones, whereas terpenes and other compounds for all the different fruits, dried species that they use remains relatively stable. And then we perform chemometric analysis for all this data that you can take from the chromatographs and we try to group the different groups. And there is some freedom here on how to group. As you can see, we don't show the whole different treatments, all different treatments that we use to develop to take the volatiles from. But we figure out that it's better to go with early infestation, late infestation, or end control. And that depends on the model outcomes when you're processing the data. Of course, for the inspector, in the end, what will be interesting is if it is infested or not. So it can be even simpler, but that doesn't mean that the algorithm will work better if it is simpler. But you will see that in a while as I go through the results. So here are the results for Ceratitis capitata, and you can see the late infestation, early infestation. They are clearly separate from the control is somewhere in the middle. And similar picture for Bactrocera dorsalis. Again, I'm talking about peaches and Bactrocera zonata as well. And if we take all together the results for peaches, so we found that there are specific compounds like ethyloctanoate and hexylacetate that are positively related to the infestation from medfly, and other compounds that are specifically related to infestation to the Bactrocera species. Moreover, these statistics show that there are certain esters that are related to fruit fly infestation, and linalool especially is related to early infestation. And we went even further to do some statistical analysis about the total compounds, the total amounts of volatiles emitted, and you didn't see here for the pitches any difference between the different fruit fly species. When you look at pears, the picture is quite different, and there are different groups that dominate the chromatograph. And you see that the main chemical groups here is terpenes and esters. And especially farnesen is one that it was found in large abundance in the headspace of pears, where there are some esters, butyl acetate, hexyl acetate, that were the main esters found both in infested and non-infested pears. You also perform this chemometric analysis for the different groups for pears. Here's the picture for dorsalis, and there are specific compounds actually that contribute to the separation between control and mature infested samples, such as linalool and methyl octanoate and others. I'm not going to say all of it. And all of this were positively rated to infested samples, whereas other compounds like FF-IPM and Limonene were positively correlated to the control ones. Similar to the previous picture, also the Bactrocera zonata, there was a specific group of compounds that our chemometrics analysis saw that positively correlated with infestation at specific days after infestation. Overall, the analysis for pears, contrary to the pitches that I was talking earlier, the total amount of volatiles emitted is influenced by the fruit fly species. So there's a different amount emitted depending on the species. And there are specific compounds that are, again, as in the previous case, that are positively related to infested pears, mainly some esters in this case. For apples, we have a similar picture like pears. Again, farnesen was found in large amounts, and esters are dominating the picture here. But there are also a small group of ethyl esters, of small carbon chain that increase with maturation during keeping the fruits in storage compartments. Again, we perform our chemometric analysis for the different species for apples, and specifically for zonata, we found 29 different compounds that contribute to the differentiation and are positively correlated to infestations. So again, there are specific compounds that are related to the infestation. And when you look at this picture, you can see that for example, the total esters emitted differ between for example, between Ceratitis capitata and Bactrocera dorsalis, and in one case decrease, and in another case increase with time in comparison with the control. Again, the total amount was affected by fruit fly species. Okay. All right, then we move forward to citrus, oranges in particular. And again, here the picture is quite different. Overall, we found 128 compounds, and 112 of them from non-infested, 122 from infested orange fruits. And here we found that fruit fly species and infestation status had a significant effect in the total amount emitted. The main compounds were Limonene and beta-ocymen and also the homotropane DM and D, as you can see here in the chromatograph, and other compounds too. This here you can see the difference from the days as the infestation and development of the larva progresses into the fruits. And some compounds, Limonene, for example, was high in the beginning, but then decreased to levels before infestation, and then Ocimene remains high even after the several days after infestation. But if you look at all together, Limonene, as explained previously, is high immediately after oviposition, whereas Ocimene remains high to the infested fruits after until the end, whereas DMNT basically increased with time and slightly other compounds as well. So I already said that the total VOCs emitted are affected by the fruit fly species and the infestation and time as well. And for citrus specifically, we have Limonene and beta Ocimene DMNT that characterize in the infestation. And Limonene in particular for the early infestation. So the conclusion for the first group of our results, which were to see which are the volatiles emitted by infested and non-infested fruits infested by the three different fruit flies, it's clear that the fruit fly infestation indeed triggered emission of herbivore-induced plant volatiles in these post-harvested fruits. And our analysis showed that it's possible to discriminate healthy and infested fruits based on these compounds. The key point was that linalool, for example, is characteristic for infestation, early infestation in peaches, certain esters for pears, decrease in farnesene in apples, and simultaneous emission of high levels of Limonene, Ocimene, DMNT, and beta-methicene characterize again early infestation in oranges. But that's just the first part. Then you have to go further and see what you can do with these results. And this results we believe that can be used for the training and validation of an electronic nose for detecting in real time the infestation by the fruit flies. So there is a potential for detection of herbivore-induced volatiles by a digital tool for pest surveillance. And an odorant-based detection tools such as various electronic noses, e-noses as they used to call them, have been developed since the 90s for a variety of commercial applications in agricultural, biomedical, environmental, and regulatory industries to assess the quality of products mainly. For instance, fruit aromas can be sampled to measure maturation and determine fruit harvesting time, or a combination of volatiles of specific concentration can be used as a biomarker. But first, knowledge of the chemical profile is needed, and this is what we have done so far, and what I presented to you previously. What is the chemical profile of the different fruits of the different infestation by different fruit fly species and at different times after the infestation as larva progresses into the fruits. And this also involves complicated multivariate statistical analysis of a complex blend. As you have seen that for citrus, for example, there are 128 different compounds. And in order to identify compounds of interest, that is compounds that vary reliably between samples and can be used as biomarkers. Now, precision and accuracy of these sensors relies on the selection of the right predictors that are specific enough and reliably occur above the limit of detection to be monitored by artificial olfaction machines. And eNoses use a sensor array that converts chemical vapors into electronic signals and create a digital pattern, which is unique and specific of the gas being analyzed. The accuracy of this sensor actually depends on the type of sensors and the statistical classification. So eNose is a tool capable of emulating the mammalian olfactory system by recognizing different odors and giving repeatable responses. We use AirSense Pen3, which is an intelligent chemical sensor array for identification of gases and vapors, which is equipped with 10 metal oxide sensors for different categories of detectable volatile substances. A few words about how we train this equipment. We again created different categories of fruits, infested or non-infested, and different time intervals from infestation by the 3 different fruit fly species. And in order for the equipment to operate and to take a reliable sample, the fruits are placed in a bag, in our case, for 15 minutes, so we have an accumulation of vapors within this bag, and then for 80 seconds we have data acquisition with the electronic nose that we use. And what you get afterwards is what you just see now on the screen, which is the response of the different sensors to the group of volatiles that are being detected. And the detection is valid when the different sensors reach a plateau. And this, for us, we use a specific period of time, and this indication is then further used in the statistical analysis to come up with some results, which I will show you right now. So before explaining what you see in this slide, the data that I just say that we retrieve from the electronic nose, which is the response of the sensors of the electronic nose, then are treated differently and to develop an algorithm. Then first there is a calibration of the data, then it's a cross-validation of the data, then find out the prediction of the model. Then you use different model based on the method of the key nearest neighborhood. And on your left, you see the key nearest neighborhood to 3, and to the right is to 1, which is, in our case at least, was more robust. And now what you see here is for the different groups of the treatments, control groups, mature fruits, or contaminated, CON2 and CON2 infested, actually, categories. And you can see that the prediction is up to 85%, top bottom right on the slide. For Bactrocera zonata, when fruits were stored at 20 degrees, we did the same thing, and we kept the fruits at 25 degrees. And then here, the algorithm that developed for Bactrocera zonata on peaches was not very robust and went only up to 65% accuracy in prediction of the infestation of the fruit category better. And we did the same in peaches for Ceratitis capitata, again at 25 degrees with 75% accuracy of the prediction. And when we went up to 20 degrees, accuracy was better, up to 85 degrees. 85% correct classification of the different fruit categories. And we went further for other temperatures. For example, here in apples, we worked at 36 degrees. After infestation, we kept the fruits at lower temperature. And there, the model was even better. And especially the K1 model reached accuracy of 88%. For that was, Bactrocera dorsalis and Bactrocera, another set of model, a different grouping. The difference between this slide is that we have a lot of groups. I said earlier that there is some relaxation here. The UK group actually infested and non-infested. So here you have all categories together, and the model goes up to 88%. And here, when you reduce it to just 3 categories, you go up to 91% of correct classification of the different fruit categories. And the same is here, again, with 3 categories for Bactrocera zonata on apples at 6 degrees. So the model worked quite well in certain categories. The overall category is 84%, but in some of the groups, it may not be so good. For example, the category day 14 is confused a lot with the control fruits, as you can see here, the red. So it takes a lot of elaboration on the different models that can be applied in order to see the results. Similar is here for Ceratitis capitata at 6 degrees, quite good overall accuracy at 86%. And finally, for mandarins, because I didn't say that before, but we also work on mandarins beside oranges. Here you can see the results at 25 degrees for zonata and capitata, with relatively robust results at 78 and 77% of accuracy. And with that, I think I reached the end of my presentation. And in summary, what I would see as a take-home message is that the post-harvest fruits do respond to the infestation and produce herbivore-induced plant volatiles. And it's possible to discriminate healthy and phased fruits based on this herbivore-induced plant volatiles. And electronic nose is a technology that is promising in detection of infested fruits by fruit flies. Although I understand that it's a quite complex phenomenon and requires a lot more elaboration of the ability of the nose to reliably detect and categorize infestation and non-infestation of the fruits during an inspection. With that, I think I'm finished. Thank you. Nikos, back to you.
Thank you very much, Panos. Very interesting talk indeed, and rich of content and rich of data and elaborated experimentation. So I think we can open the floor to questions or remarks to Panos, if there are any from our attendees. I cannot see anything on the chat, to be honest, but Panos, I will start with what you said last about, the challenges on going from the lab and all of this elaboration, going to the real world. And I would like just to comment on how far we are from having a system that can go to the operation, to places that, fruits are arriving, and do the inspection and assist the phyto inspector there.
I think, Nikos, that this is a question that should be answered not only by a group like us, but also by engineers as well, because what we try to show is that there is indeed variation in the volatiles emitted in the different categories. And ideally, you will have, when I start talking about the electronic noise, I mentioned that you need to develop specific biomarkers. And what you use as an electronic noise is for general use. It's not specific. Perhaps the answer to your question is if we can have a specific developed electronic nose with specific sensors that have been structured and developed and constructed to detect this specific group of volatiles. And you have, when you're doing the inspection, you have to know have to know something about your sample, which is, what you're looking for. But I think the key is to have a sensor which is sensitive enough to find the the differences, because the differences are there. And that's a big problem. If we were able to have an TCMS in the inspection, then there would be no problem. Like we can see the difference. But we can. It's laborious, cost, and it costs a lot, and you need a lab.
Okay. Any other comment or question for Panos?
Just received, I think, a question from your Gazette. What was the max volume of fruit you tested?
There was an interruption. I didn't hear you, Georgia.
Sorry. What was the maximum volume of fruit you tested?
Less than 500 grams. We try to keep it the same for the different fruit categories, actually. So that's why in some cases you have one fruit, some other 3 fruits, depending on their size. But it was relatively, below 500 grams. You know, I didn't say that. I thought it could be a question. When you are using the electronic nose, we use these bags to pack. And in practice, this what we will do now is we will go and we will wrap a carton with fluids, and we will take the volatile samples from that enwrapped carton. And this would be actually the procedure in place if ever goes to the real world. For the inspector to take a carton of fluids, wrap it with something like we did, and then take the air sample from there and see if it's positive or not. And if it's positive, then that, of course, at the beginning you need to verify that it is positive and find the fruit fly, because especially for the inspection for carding purposes, you need to identify the species as well. And what the machine can speed up the procedure for reaching a conclusion that the commodity is infested. And so this is something that you can we will do in the near future. And this is how the inspector will use it in practice. And actually, and this is how it's used, because when they are used for commercial reasons, as I said, for regular inspections to see the quality of a product. So at this time, this specific equipment, this is how it is used. They wrap even a pallet, and they take a sample from air of the pallet to see if it's in one category or another.
Panos, another thing is, what is the temperature dependence of the sensitivity of volatile detection? Is there a specific range of temperatures that you can collect more efficiently the odors, or this goes across broader areas of the spectrum?
Well, I see. You saw that we use different temperatures, and the model works better when the fruits are maintained in a lower temperature. But in practice, the inspection will be done in a working environment, let's say 20 degrees or 25 degrees, in a room, in an inspection room, as I imagine. And there, I don't see much of a problem with the temperature. Usually, at least the nowadays commercial noses and sensors, they have a problem with the humidity. And humidity may interfere in the results. We didn't specifically test it, but from the literature, you can see that one of the problems is moisture and humidity in the air that may interfere, and not so much of the temperature. We use the different temperatures to prove that the differences exist, no matter if the fruits are kept at 6 degrees or 20 degrees or 25 degrees. There are differences in the volatile blends emitted by the different fluids.
Okay, actually, because I cannot see, okay, there is something, Georgia, from
We just received another question. Practically, you should aim for in-nose to inspect fruit in the truck load just before the packing process, rather than in carton boxes. I think it's a remark, not actually a question, if you can comment on that.
It will be ideal. I can say that it would be ideal, and you have think about that. But think of the complexity of a factor. Yes, for the exported, that would be ideal. For the imported, what they do is to inspect the carton box, and at the end, all National Plant Protection Organization and inspectors, both at the export and import, they check the level of cartons. I'm not very optimistic that at the moment that there's technology to identify inspection during the truck, in the truck. There are some techniques, but not like that are still at the very early experimental level, as far as I know.
Okay, and another question from Costa Zarpas. How do environmental conditions affect in-nose efficiency?
Yeah, I said about that it's mainly the humidity that affects the efficiency. Generally, they're quite robust. As if you remember the picture, they are a compact machine, very close from the environment, and only has to take for a few seconds the air sample, and then to process this sample very quickly. Of course, in every sampling, there is a procedure to clean the equipment, but again, that doesn't take long, just a couple of minutes.
Good. So I cannot see additional questions. We can discuss with Panos for hours on the subject. We did it already, and we definitely can continue. This is a really challenging topic with various factors coming into play. But because, as you all know, in Greece and in other places in Europe, this is a very hot day. So I would suggest to close our discussion now, to thank Panos for the very comprehensive and the very nice presentation. We know that there is a lot of work to be done, but there is also lots of achievements by the group of Panos and the Benaki Phytopathological Institute that makes our project really happy. So Panos, thank you very much for this nice talk. And for those of you who are still with us, stay tuned and follow our announcement for the next webinar. Thank you very much. Thank you all.
Thank you, Nikos. Thank you, everybody. Thank you, Georgia, for organizing that.
Thank you, Dr. Mylonas, Professor Papadopoulos. Of course, more announcements. You can check our website and our social media pages for whatever our project is heading next, and you can find out there. Thank you very much. Goodbye.
Georgia, thanks a lot for all
of you.
nice energy.
Thank you. Goodbye.
Bye-bye.